the soybean pathology lab. Well, we're very happy to have you here today. We're very happy to share a little bit about the results we got from 2023 and getting ready for our 2024 season. Planting considerations, especially when we are going to decide to maybe plant earlier than common. One of the things we have been working and collaborating with Dr. Laura Lindsay is in the Battle for the Belt project, where we have 5 different planting dates. And really here, what we're looking into is that disease triangle that we talk when we want to describe when disease can happen. Pathogen, a virulent pathogen, a susceptible host, and a conducive environmental condition. Now, when we're dealing with soil-borne pathogens that inhabit the soil, really changing the environmental conditions can enhance certain groups to be more aggressive, to cause disease over others. For example, if we have saturated soil, a very wet spring, water that is not well drained sitting in the field, some of the water moles, such as Phytophthora and Pythium, can become very aggressive and cause disease. In other situations, when we plant early and we have very cool soil conditions, certain organisms like Fusarium virguliforme that can cause sudden death syndrome can infect more readily. And therefore, eventually in the season, we may see more symptoms of SDS. And like that, we may have different other organisms. So very important to know the history of our fields to be able to determine the risk that we are dealing under different conditions. So what I want to do today is I want to invite Jenna Moore, a PhD student in my program, to share a little bit about the results that we have been gathering and a little bit of the understanding that we have been achieving with this project.
Hi, I'm Jenna Moore. I'm a second-year PhD student in Dr. Lopez-Nicora's lab. I'm working with Taylor on the Battle for the Belt project. We're working on the pathology side. For this, we're using 2 different approaches. A conventional approach, where we are plating the seedlings in the root rhizosphere soil. And then we're also using a metabar coating approach, where we're taking soil and we're going to later extract DNA from the seeds, and it tells you the entire community in a single sample. So instead of just using molecular identification to identify a single species, this will tell you how many species are in the entire sample. From last year's data, we saw different community structures based on different planting dates. We also saw different community structures based on crop, showing that certain fungal pathogens have a preference for different hosts. In addition to host specificity, we also saw differences in location. So there's going to be different fungal communities based on things like soil properties and weather conditions in different locations throughout Ohio. We're looking forward to repeating this again in 2024. We have our first planting date from Western Agricultural Research Station here, and this is the conventional approach where we are doing isolations directly from the plant material.
When Jenna talks about different community structure, last year we experienced in one of our planting dates a drought spill. We had a moment where conditions were very dry, and we saw more recovery of certain organisms that do very well under these conditions. For example, Rhizoctonia. On the other hand, when we look at more wet conditions or cooler conditions, we start seeing other organisms, and that's what we are trying to look. And not only observe these in a very conventional way and traditional way as growing these organisms in a plate, but also sequencing them. So not only Jenna can characterize these morphometrically, morphologically, but we also extract DNA and then we confirm their identity with molecular techniques. This will also allow us to explore more organisms that we cannot isolate very easily in a plate, in a medium. By sequencing everything that we have in the surrounding soil of the seedlings, we will be able to explore more these different communities and associate them to the different environmental conditions. This brings us, of course, with a message of the need for scouting, right? Walk your fields. After it's planted, a week later, 2 weeks later, when the plants are starting to come out, look at what's going on. If there's anything that resembles not healthy or poor growth, it will be a good idea to pull those samples from the field and send it to a lab. Here at Ohio State, in the Department of Plant Pathology, we can help with this type of diagnosis. You can send your sample to the soybean pathology and hematology lab, and we will be very happy to help identify any type of problems. We also talk about planting early and in cool seasons that can allow certain organisms, pathogens like the one that causes SDS, to be more aggressive, to infect and cause more severe SDS symptoms later on in the season. So if you do see SDS symptoms in the field, we encourage you to send those samples to the lab. As a matter of fact, in addition to what we're doing with the early planting, Jenna is also working on a project when we are trying to understand that diversity of the fungus that causes SDS in Ohio. We're doing this with funding from Ohio Soybean Council, and we ask you, if you see SDS in your field this season, pull the sample with the root system and send it to the lab. We will be able to isolate the fungus and further evaluate that diversity, both in pathogenicity, how aggressive this can be on soybean, but also if they are showing resistance to fungicides. And with that, this leaves us with the message of, well, what do we do now? If I know I have pathogens in my field, if I know that certain environmental conditions are not under my control, what can I do to protect my crop? Resistance is the best way to do it. Select varieties that are resistant to pathogens that you may have in your field. Seed treatment is another way to protect our crop health. This seed treatment has to be specific for the type of pathogens you have. So the water molds such as Phytophthora and Pythium are not true fungi. So specific products are meant to control those. Fusarium, Rhizoctonia, and others that are true fungi will be controlled and managed with true fungicides. So know the products according to the threat that you have in your field. Cultural practices such as well-trained fields that always will help with avoiding compaction and saturation will always help our plants not only to grow better, but to avoid becoming more susceptible to these pathogens. And with that, we hope you have a great 2024 season, and go soybean.